Hello everybody, welcome to that webinar in which we will talk about which are the critical points during fertilization and which will be the barriers that sperm should overcome in the female tracts in order to reach this oocyte fertilization. But before starting, I would like to refresh what is a sperm, okay? So the sperm cells are male haploid cells that are very specialized in fertilizing female gametes. This is very important because these cells have evolved for a long period of time ago just to reach the oocyte and overcome the different barriers found in the female tract. Okay, and this will be critical for predicting the success of fertilization when we are working in a laboratory. Okay, so these sperm cells are generated in the testicles through a process that is called spermatogenesis. This process specifically, these latest stages in which sperms acquire the final shape, the final morphotype that is characterizing these gametes are very sensitive to external factors, smoking, drinking alcohol, the stress, the temperature of the testis is affecting a lot on these sperm features and will provide you from very valuable information about the health of the patient. So apart from this morphological information that we can extract from that cells, also is important to take in mind the functionality of these gametes. This functionality is acquired specifically during the epididymal maturation, okay, in which these immature sperm cells are acquiring a lot of changes in the membrane composition and is acquiring these new properties for acquiring the motility. Okay? So these sperms that are now functional and morphological mature should be mixed with a specific secretion from the accessory glands, which will confer the nice background that will help this sperm to overcome the first barrier on the female tract, that will be the entrance to the vagina. Here, the pH of the vagina is very acidic, so sperms acquire motility only when there is a pH slightly basophil. So it's important to reach optimal accessory glands, this plasma seminal little basophil, in order to compensate the acidity of the female tract and allow the motility of the sperm cells once entering the female tract. In the human field, we are very lucky because we have a very clear rules about how to evaluate the quality of an ejaculate. That is the WHO guidelines for semen analysis. Just to refresh, just once we receive a human ejaculate, it is recommended to allow for liquefaction. This process is very important to do it in order to then perform the macroscopic and the microscopic evaluation. There are some discrepancies between laboratories about which is the best way to allow liquefaction, because WHO recommends to do it at room temperature, but there are some laboratories that prefer to accelerating this process at 37 degrees, okay? In that case, there is not a best way to do it. It will depend, each laboratory, each technician should decide depending on the priority of the analysis to do. For instance, if you're receiving a lot of samples at the same time and you don't have time enough for evaluating motility, vitality, these parameters that are very sensitive to the passage of time immediately, you can just allow liquefaction at room temperature in order to maintain the metabolism of these sperms and from the enzymatic proteins that allow the liquefaction. and just preserve this sample for some additional minutes, okay? Conversely, when you leave the sample in the incubator at 37 degrees, you are activating, you are accelerating this metabolism in the ejaculate. So uh, also you will be activating and increasing the percentage of reactive oxygen species on that sample. So if you are incubating at 37 degrees, immediately you should start the motility analysis. Okay, if it's not possible, just leave to complete the liquefaction at room temperature. Okay, so when you are doing the macroscopic examination, this is a very important process that normally technicians tend to forget, but it's very valuable because it could provide a lot of information about if there is the presence of blood in the ejaculate. You can easily evaluate it through the coloration. Okay, when there is pinkish, when there is reddish, it could indicate the presence of red blood cells in the ejaculate. Also, when you observe a lot of crystals or particles, could indicate a possible contamination with urine. Okay. Also, it's very important the volume. According to WHO, normal samples are assumed as these samples that contains a volume from 1.5 to 6 ml. What happens when the sample is below this 1.5? You need to consider the option to found a retrograde ejaculation or maybe an obstruction on the patient. So you need to come back to the anamnesis of the patient and just try to evaluate if there is any real problem on the male tract that could difficult the complete ejaculation of the sample. Okay. The viscosity is another critical point, not under a clinical point of view, but it will be useful for technicians in order to predict the difficulties that will arise during the semen analysis, because viscosity will difficult the assessment of motility, of including concentration, morphology. So samples should be handled on a different way when they are very viscous. Okay, and obviously the pH will give you from very valuable information about the effectivity of the accessory glands. Okay, if there is an equilibrium between the prostatic secretion and the secretion from the seminal vesicles. Okay, this pH should be slightly basophil. This is important in order to contrast the acidity of the female tract. Okay, just refresh. Remember that sperms are only motile on a pHs that are partially basophils. Concerning microscopic information, WHO recommends first of all assessing motility because it's very sensitive to the passage of time. When this motility is very poor, is below this 40% of motile cells, it is recommended to do vitality in order to evaluate how many of these immotile cells are alive and how many are dead. Okay? Obviously, the concentration will be very valuable to predict which will be the statistical probability to ensure fertilization. Okay? And morphology. Okay? Also very informative to evaluate the health of the patient. The problem relies in that we have so many cases in which the spermatocytograms are normal. There is not a clear male factor after doing these common assessments. Then we evaluate the female and there is not a severe problem in the female, but fertilization is not occurring under natural conditions. So what happens? We have more than 30% of couples diagnosed as idiopathic infertile. Okay, so normally you will find a lot of publications that discuss about this fact. And normally these cases are related to, in some cases, problems in the hormonal balance. Okay, they're normally related to a deficit of testosterone. Okay, alterations in reproductive physiology, or well related to immunological problems, the presence of antibodies, specifically when there is a breakage of the blood barrier in the testes. Okay, but we will focus special attention to the alterations in the reproductive physiology, specifically to the alterations in the sperm physiology. Okay, because there are so many studies that have been recently published that have revealed that just evaluating the typical parameters like motility, vitality, concentration, or morphology is not enough for explaining which is the problem with the male. So this information is not enough, and we need to go further on that. And this is when the functional tests are acquiring more and more and more importance nowadays. We will talk about these functional tests that are evaluating the biological functionality of sperms, okay, to react to specific barriers in the female tract. Okay, so just paying attention to the process of fertilization, we know that once sperms enter to the female tract, the first barrier that encounter is the cervical mucus. Okay, this cervical mucus will be probably the most critical barrier because we'll select for a specific profile of sperms. Okay, only these sperms that are able to overcome that barrier will be able to arrive to the fallopian tube and therefore just reach the oocyte, penetrate the layers, and then ensure fertilization. The problem is that this is not the end of the story because there are a lot of publications that revealed the necessity to test for the integrity of the DNA because this DNA will have a very important effect on the embryo development. So if there are some damage in the sperm DNA, all this fertility process, all this fertilization process could fail. Okay, so it's very important to assess it. If we are going deeper on each critical point, we will just we have to think about sperms. We are sperm. We are entering in a female tract, and the first barrier that we'll encounter is just the cervical canal. Here we will find our cervical mucus that generates like a net that will difficult the penetration of a high percentage of sperms that enter there. So this cervical mucus will first do a specific selection for vigorous sperm. Specifically, only these sperms that are swimming pretty fast, faster than 25 microns per second, and that follows a progressive motility, will be able to penetrate this layer. If there is one sperm that is swimming following circular tracks, will never cross this huge barrier. Okay, so will be retained there. Also, this cervical mucus will be very important for selecting sperms according to its morphology. All these morphotypes that are very abnormal, these macrocephalias, double heads, or just piriform heads, will be retained. Most of them will be retained there. So it is at the same time that selecting for motility, also selecting for morphological features on the sperm cells. Okay. There is one very important point concerning morphology because If you pay attention to the cutoff that they are normally used according to WHO, it says that a normal sample is a sample that contains a 4%, at least a 4% of sperms assumed as normal. It means that the 96% of sperms are abnormal. So this is statistically something that is not very significant. It is difficult to predict the success on fertilization, just taking into account that the human spermatogenesis is so poor, it's not so effective, it's generating a huge quantity of abnormal sperm cells. This is why it's very important just to assess not only for the normal percentage, but also assess the types of um, abnormal cells ejaculated. This is why WHO recommends to follow this abnormality index, the teratospermia index and the multiple abnormality index. In the last edition of the guidelines, it has published this reference cutoff in which, for instance, for the teratospermia index that we found in fertile couples, we are reaching values from 1 to 1.5. Okay, it means that there is only one or maybe two structures of the sperm cells that are abnormal. Okay, we expect to find ejaculate in which there are not so many abnormalities in the head or in the midpiece, in the tail, or in the cytoplasmic droplets. Okay. Conversely, when there are problems on fertility, these teratospermia index are reaching the value of 1.8. So this is indicating, it is suggesting that the success of fertilization of a couple in which the teratospermia index is 2 or is above 1.8 could be more difficult than for another couple with a teratospermia index below that value. This is why it's important just to compare which is the percentage of normal shapes and which is the level of damage within the abnormal ejaculate, okay? And just to predict which will be the success, because in some cases we can find patients with the same percentage of normal shapes, but with a very different teratospermia index. And in these cases, the probability to reach the oocyte will be different for these patients despite having the same percentage of normality. Okay, so just we need to think about and pay attention on using that type of indexes to weight the level of damage within the abnormal part of the ejaculate. Okay. Apart from this motility and morphological selection, additionally, this cervical mucus also allows the capacitation of sperm. This is a very, very important process because we have to think that when sperms are maturated in the epididymis, are covered, the surface of the sperm is covered from a lot of proteins, carbohydrate, and cholesterol that will inactivate these sperms, that will generate like a cap that will prevent the premature acrosome reaction meanwhile it's crossing the cervical mucus, or just preventing from the hyperactivation or detection, the premature detection of the oocytes before acquiring all the metabolical changes that need to be submitted before reaching the oocyte. Okay, so the cervical mucus is very important just to allow the capacitation. But how is possible this capacitation? We have to think that here, okay, the capacitation consists in two types of events. There are some fast events in which the sperm easily react. Okay, when you incubate the sperm cells with carbonates or calcium, automatically this optimal sperm should activate the motility, should start a vigorous motility like could be typical from the hyperactivation. Okay, but capacitation also is defined by some slow events, that are dependent on the presence of cholesterol acceptors. That could be the albumin. Okay, so this is the key factor during penetration through the cervical canal, because the cervical mucus is full of albumin that will act as a receptor of the cholesterol cap and will detach this cap, like you can see here. All this cap, all this cholesterol will be trapped, will be captured in this matrix, in order to that when sperms reach the fallopian tube, they are lost. They have lost this cap and now are sensitive, become sensitive, present all these receptors that will allow the perception of the oocyte are discovered, okay, are exposed to the surface of the membrane. This is when we say that the sperms are capacitated. Capacitated means sensitive, with the receptors exposed. Now the sperms are able to perceive the chemical stimulus from the oocyte. Okay? So this is a very critical process that we have to take into account, specifically when we are working in the lab. How can we simulate all this natural process that occurs in the female tract? Imagine that you receive one ejaculate, we evaluate the motility, it is not very good. Morphology, it is difficult to predict in some cases. There are so many abnormal shapes, and we want to use that sample for doing a reproductive treatment. How can we prepare this sample in order to do this insemination or this IVF or this ICSI to the female? Very easy. This process can be reproduced by using a density gradient, a Perkol gradient washing method. Okay? This method, this typical, is normally used, and we normally forget why we are using this type of procedure. Okay? This, when you are doing a Perkol gradient, density gradient, first, we are separating the motile part from the immotile part through the parts within these layers with different density. The sperms with greater motility will be precipitated to the bottom, and therefore will be selected and found in the bottom of the tube. Meanwhile, those abnormal sperms, these sperms with abnormal morphologies, will have a different density than these normal sperms, so will be retained on the upper layers. So at the same time that the gradient is selecting for motile sperms, are also selecting for morphological normal sperms. Okay, so all these sperms with a greater level of abnormalities will be discarded with this method. But another important point to take into account, this Perkol gradient, when it's enriched with albumin, it will also allow the capacitation of sperms. Okay, so also will retain, while these sperms are moving through the different layers, these sperms will be losing this cap, and therefore these recovered sperms will be capacitated. Okay, this is the most easy way to simulate this natural process in the lab. Okay, so let's come back to the natural process. Once this capacitated sperm reach the fallopian tube, enter to the uterine tube, here there is a critical point, that is the synchronicity with the oocyte, with the arrival of the oocyte. This is important. Why? Because these capacitated sperms that are now sensitive can perceive the presence of the oocyte, will arrive to the tube and listen if there is any chemical stimulus from the oocyte that is close to them. Okay, this is a chemiotactic behavior. In that case, these sperms, if are not able to perceive anything from the media, if they are not perceiving the presence of the oocyte, they will attach to the walls and wait until perceiving this movement. This is why it's very important to reach a good synchronicity between the oocyte releasing and the moment of insemination, or just to do the treatment. Okay, so these sperms, once perceive the chemical arrival from the oocytes, will change their metabolism, will change their behavior. They become hyperactivated. Hyperactivation is a typical behavior that occurs when these sperms are incubated with a stimulating medium, with a medium that is rich in calcium, that is rich in hormones, okay, like could be an enriched heterotubal fluid or some IVF media, okay. This stimulation will stimulate the chemical stimulus from the oocyte, and therefore will start this crazy movement of sperms that is called hyperactivation. Here sperms start swimming faster than 150 microns per second. Okay, they start swimming very fast, but are following a lateral movement of the head that is very wide. This movement is very important, especially when they are attached to the fallopian walls, because it will help these sperms to detach from the walls and swim so fast through the oocyte. Okay, but this hyperactivation behavior, it is characterized by two main stages. This first stage, in which sperms are activated and start swimming so fast, following a clear progressivity through the oocyte, but when they are very close to the oocyte, they start moving like that. It shows a typical drug that is called star-like movement. They lose their progressivity, okay? And this is a very important point to consider, specifically when you are selecting sperms for doing an IVF, because technicians tend to select only for these sperms that are swimming fast, but also progressively. But we have to think that these sperms that are swimming like that, without any progression, but they are swimming very fast, it means that have reached the latest stage of hyperactivation, have completed the process, okay? And therefore should be also considered for selecting this sperm for doing the IVF, okay? Then, when these sperms reach the oocyte, they will contact to the two external barriers that are protecting the oocyte to prevent the polyspermia, okay, to prevent the penetration from multiple sperms. In that case, there are two main oocyte barriers that are the granulosa cells and the zona pellucida. Both are very critical points because at very selective stages for specific profiles of sperm. So, when the sperms reach the granulosa cells, these granulosa cells are attached to each other by through hyaluronic acid. Okay? When sperm bind to this layer, only these sperm that are able to recognize that layer and initiate the translocation, the exposition of the receptors for the hyaluronic acid, will be able to break these chains of hyaluronic acid in between the cells and make the whole. Okay, so here is very important to assess how many of our capacitated and hyperactivated sperms are able to penetrate the granulosa cells. There are so many tests that we can perform, like the HbA, the hyaluronic acid binding test, in which we can evaluate how many sperms are able to break this layer, or how many are able to recognize this specific binding. Okay, so once these sperms are able to penetrate, to separate the cells, and together with the combined movement of the tail, are able to reach the second barrier that is protecting the oocyte, that is the zona pellucida. Here, there is a very specific recognition between the sperm and the oocyte. Only these sperms that are able to bind, there are some uh, receptors on the membrane of the outer membrane of the sperm cell that can bind to the zona pellucida. Only when there is a binding of these receptors with the zona pellucida, it will trigger a change in the metabolism of the sperm cell and will trigger a calcium influx. Okay, this calcium influx will involve the fusion of the plasma membrane, okay, this plasma membrane, outer membrane, with the outer acrosomal membrane. So this fusion will trigger the release of all these proteins, of all enzymatic proteins that are contained inside the acrosome vesicle. Okay, this process is called acrosome reaction and will allow the chemical digestion of the zona pellucida. Okay, also this process is very important not only for penetrating this layer, but also for exposing all these proteins, all these receptors from the equatorial segment that will allow the recognition of the plasma membrane that is surrounding the oocyte, the oolemma. Okay, so these sperms that are not able to reach the acrosome reaction will not be able to penetrate the zona pellucida, but also will not be able to recognize the oolemma because these proteins from the equatorial segment will not be exposed. Okay, so unfortunately, this is not the end of the story. Why? Because now we have one sperm that have penetrate this layer that can fuse with the oolemma and start the embryo development. But what happens if the DNA of this normal sperm, optimal sperm, is damaged? All process will fail because the DNA has been related to a miscarriage on the embryo development. Okay? Actually, in close to 30% of cases, diagnosis of idiopathic infertility are related to high levels of fragmented DNA. Okay? This fragmentation is very linked to the presence of oxidative stress in the patient. Okay, so this oxidative stress is also related to some environmental factors that are affecting to our patient, like could be the tobacco, the alcohol. At the end of the presentation, you will find some bibliography, some reference that you can just read in order just to evaluate which could be the different factors that are affecting a lot on our sperm features. Okay, so it's very important for the generation of the pronucleus, the condensation of the pronucleus, and fusion of the gametes of the pronucleus. Sorry, just to guarantee a good quality of the sperm DNA. Okay, so this is a critical point that we need to assess specifically before doing any reproductive technique, because everything could fail just because of the presence of DNA fragmentation. Okay. So after having all this into consideration, do you think that just doing a conventional sperm cytogram in which we are evaluating motility, vitality, concentration, and morphology, do you think that is enough for evaluating which is the problem of our patient, which is the influence of the male factor on this infertility of the couple? Okay, this information is not enough. It is important to test for the ability of the sperms to penetrate the cervical mucus, to hyperactivate, to evaluate the sensitivity of the sperms to penetrate the zona pellucida, or just evaluate the integrity of the DNA too. Okay, so on the same way that every day we are increasing more and more our knowledge about the different factors that are affecting on our sperm cells, also the ways to analyze these different parameters, these different factors are evolving too. This is why the incorporation of CASA system is helping a lot to our technicians, to our laboratories, to get a more accurate diagnostics, to incorporate all these additional tests that will enrich, that will help the doctors to reach a good and accurate diagnostic for each couple. Okay. In the case of microoptic, we have developed two different systems, the SCA and the SAE scope, that allows not only doing the conventional uh, uh, parameters that are recommended to do according to WHO, like could be motility, concentration, and vitality. Also, it allows to test, for instance, when there are so many round cells, you can do a specific test for peroxidase positive cells. Okay, so you can do a specific test for detecting activate leukocytes that could be linked to the presence of an infection. Okay, also these CAS systems allow to evaluate morphology in a very accurate way, and not only evaluating the percentage of normal shapes, also provide from very valuable information about which are the frequency of each type of abnormalities, in order that doctors could link each abnormality to a specific pathology. Okay? Also automatically calculates the teratospermia index, multiple abnormality index, all these index that will help you just to weight the level of damage within the ejaculate. Okay? Also allows to incorporate the functional test like the ability of sperms to hyperactivate. Within my ejaculate, how many sperms will be able to penetrate the cervical mucus, and then once stimulated will be able to hyperactivate. Okay, it's very easy to do this type of test with CASA system. When you are doing the manual assessment of motility, it's impossible to evaluate or to know which is the exact velocity, which is the progressivity of its sperm. Okay, so this is something that we have to take into account. Also very important, we are incorporating the functional tests like the acrosome reaction, the sensitivity of sperms to premature react or to be insensitive to react when contact to the zona pellucida. Okay. Also will provide from very valuable information about which will be the procedure that it is more recommended for each patient. If there is one patient that is very insensitive to undergo acrosome reaction, it's better to go directly to ICSI. Conversely, if this sperm, if this ejaculate is responding properly, maybe with an IUI could be enough. It's not necessary to go on a IVF or anything else. Okay, so it's important just to take in mind uh, all these analyses for getting a better treatment for each male. Okay, and obviously, finally, the DNA integrity that is very valuable for predicting the success of embryo fertilization. Okay, so apart from doing all these tests, also it allows to collect all this information together on a very sophisticated report that you can customize depending of your necessities. You can incorporate some graphs, some images, the logos, or just information that you need in order to maintain the traceability of all the analysis and show the results on a better way, on a standard way. Okay, so thanks for everything. Thank you so much for your patience. And if you have any doubt, anything, you can just read this reference, okay, in which we have page for doing the presentation too. Or anyway, if you have any question, anything, you can just write me to nuria@microptic.com. Thanks a lot.